In this video, we want to see the process of germination. Okay, the life cycle of a bean plant. So a mature bean plant produces seeds and which are always housed into a bean pod. So although this one is dry, but you can really observe that this is the bean pod that we are talking about, and the arrangement of seeds within the bean pod is like as in this way. So you find them being housed into this bean pod like in this way. So when the bean plant matures, the point reaches where it dries up, and as it dries, then the seed pods also lose strength. So after losing their strength, then they split. After splitting, they release the seeds which are always housed inside of them. And after releasing them to the ground, sometimes when the garden is planted by the gardeners, these plants are harvested and dried and then either stored for further planting or else they are eaten. But then when it is just a self splitting, okay, when the seed pod splits itself, then the seeds fall on the ground. So after falling onto the ground, uh, in this experiment, I potted some of the plants. So some of the plants are potted. So, and by potting, it just means the planting of seeds into a pot. Okay, I'm terming these ones as pots. This one, yeah. So when these seeds are put into soil, like in this one, then you add water because we expect factors that favor the germination to be available if you pot a plant. Like in this case, there is soil, there is warmth, there is oxygen supply, there is water also added. So when this seed is put into the soil, then this coat, the outer covering of the seed, which is called the coat, housing the cotyledons which are inside the seed, uh, it gets hydrazed when it is in the soil by the absorption of water. And the absorption of water in this case is in two processes. That is imbibition, which is against the colloidal nature of the seed. The food substances inside the seed favor imbibition, that is water movement into the seed according to its colloidal packing of the food substances inside. So, so the other process is osmosis, and by osmosis we have got there is a very point here. Although with naked eyes you cannot observe that it is opened, but then water enters into the seed by one osmosis and imbibition. So after entering into the seed, the the cotyledons, which are food stores of a bean seed, become hydrolyzed. These ones, these ones become hydrolyzed. So, and by hydrolysis, we expect them to be broken down into glucose by respective enzymes. So, as water enters into the seed, then it activates the enzymes required for the hydrolysis of this starch into glucose. And it is glucose which is translocated into the embryo, this embryo, the growing region of the plant. Okay, it is the very active part of the plant during growth. So, so when that glucose is translocated into the embryo, then we expect oxidation. That is the breaking down of glucose into the energy using oxygen, which that energy in form of ATP is used for the growth. Then we expect immediately that at the seventh week, at the seventh day from the time of planting, we expect the radicle to emerge first. Yeah, we expect the radicle to emerge first. Are you with me, ladies and gentlemen? we expect the radicle to emerge first. And the radicle is that white. Okay, it is somehow white in color, but although this one it is mature, it is not a radical you wanted, but it is also thread-like white in color. By that point, when it leaves the seed, it is white in color and thread-like, although this one is mature. So, so this radicle blows down the soil, want to absorb water and nutrients required for the growth of the plant seedling. So after the radicle has emerged, because of this epicotyle, because we can find that in a seed we expect to be having the epicotyle and the hypocotyle. So in epigene germination, the epicotyle grows grows very faster than the hypocotyle, in that the epicotyle first pushes up the shoot system. Well, the growth of the epicotyle in epigene germination is faster than the hypocotyle, in that the shoot is forced to leave the ground instead of remaining into the ground. That is why you find in epigene germination seeds are always uh, seeds are pushed out of the ground, while in hypogene germination, the seeds remain deeper into the soil because of the hypocotyle and epicotyle actions. So, so, so after the radicle has emerged, the following one is the leaves, and the leaves are the first two leaves of the bean seed, or of all epigene germination seeds which undergo epigene germination. By epigene germination, we mean we just mean where you find seeds grow up the ground. It is epigene germination. So in epigene germination, the first two leaves are called these cotyledons. They become specialized into first leaves. But however much, these first leaves are not the typical leaves of a plant. Because the typical leaves of a plant, they are like these ones. Yes. So if you can see, these ones are just the coleoptiles. We can define them as coleoptiles for easy understanding. Yes. So so these first leaves, they absorb sunlight. Okay, they have got specialized cells. Those are photosynthesizing cells which can be used to carry out photosynthesis, that is generation of food required for the firmness and growth, continuity of growth of the plant. So other leaves, so after this